Hi everybody, welcome to today's seminar. I'm Malcolm Colley, one of the organisers of the IBH Seminar series. And before I hand over to Luke, who's hosting today's talk, I just wanted to let you know there's a couple of options. If you want to ask questions, you can pop questions in the chat and we'll pick them up after the presentation. Or you can also virtually raise your hand and I'll unmute you and you can ask your own questions. And obviously, hopefully the seminar will be available online for you to check out on our YouTube channels, on the usual kind of channels. So have a look out for that. We'll let you know the link on our social channels. So I hope you enjoy the talk, and I'll hand over to Luke now to introduce today's seminar.
Okay, today we're delighted to welcome colleagues from Australia. This is a double header. We have Professor Michelle Colgrave, who's Professor of Food and Agricultural Proteomics and the Chief Investigator on the Australian Research Council Centre of Excellence for Innovations in Peptide and Protein Science, CIPS. And we also have Dr. Clare Alone, who's a research fellow in serial proteomics within CIPS, and her research has been largely based on grain quality in barley and oats, and she'll be presenting some of her research. There'll be two presentations. Professor Michelle's initially is hoping to join us later, but her presentation is a virtual one initially, and then Claire will give her presentation afterwards. So hopefully, I think this is all in your hands, Claire, both of them. So I'll let you take the floor.
Okay. First, I'd just like to say thank you on behalf of both Michelle and I for the opportunity today, and thank you to everyone for joining online. So I'll just present Michelle's now.
Hi there, my name is Michelle Colgrave and I'm Professor of Food and Agricultural Proteomics at Edith Cowan University. I'm just going to provide a bit of an introduction to our group to explain the type of research that we do at ECU, and then you'll be hearing from Claire, who was a PhD student in the lab and has now moved on into a postdoc role. So our group really focuses on proteomics and multiomics for discoveries in food and agricultural domain. We apply primarily mass spectrometry as our key workhorse in the lab, but to understand the proteins and peptides that exist in nature. And we're also using proteogenomic and bioinformatic workflows to uncover those proteins and peptides, and then applying systems biology to understand how they function. We look to identify both the good guys, bioactives in bioactive peptides in crops and food products, as well as some of the bad guys, things like food allergens, antigens, and other anti-nutritive factors. And we do this with an industry lens. We're thinking about how do we use these discoveries to inform market development for breeding programs or to explore new treatments for, say, insects or pests. And so it's really thinking about the applied side of science. We focus on many different grains within the lab. Here you can see some of the examples of work we do in wheat, thinking about some of the quality characteristics like proteins like gluten that exist in the grains, but also thinking about some of the conditions that affect wheat, such as late maturity alpha amylase and pre-harvest sprouting, that can be a problem in terms of quality. And then we're also looking at the effects of things like drought stress or nutritional stress as we move into a ever-changing climate, and we're trying to maintain productivity while also maintaining the quality of those grains. And as I said, we also start to look at things like the bioactive peptides and the proteins that are involved with celiac disease or food allergy. And as you'll see on many of these slides, I've listed many of the collaborators that we work with. This is an example of barley, and you're going to hear a lot more about barley through Clare's work, which was really looking at malting quality. But we've also considered how different grains impact on beer quality through either flavour proteins that are involved or those that are involved with things like haze formation or foam stability. And then we've also been interested in looking at some unique varieties of barley that have different colours and they have different antioxidant profiles. And again, looking for things like bioactive peptides. And much of my early work in collaboration with CSIRO was about developing a low gluten barley, and we used proteomics as the tool to ensure that we were selecting the lines that were lowest in gluten and then could be taken forward in the breeding programs. Oats is a more recent interest of ours, and we've had some great success with our collaborators and partners across industry and international partners. We've been involved with the oat genome, and this was the work of Angela, who has in my lab primarily, but also with Paul Bose. And through that, we've been able to map the gluten proteins or the gluten-like proteins within oats and understand the difference between the epitopes that are expressed in oats versus what you see in traditional cereals like wheat and barley. We look at those health aspects. We also have PhD students looking at how the protein profile of oats affects product quality, and in this case, we're looking at oat milk. And we also, again, look at things like nutritional stress and the use of fertilizers and how that impacts on the final proteome that is expressed. We extend beyond cereals into legumes, and so here's a few examples. Sweet lupin, which is a really nutritious, some would call superfood, but also contains some allergens and anti-nutritional factors. So we use a combination of proteomics and metabolomics to be able to explore both those small molecule toxins as well as the proteins that can be responsible for allergy. And that enables us to select lines that have lower allergy potential and also have the sweet variety, which is essentially lower in alkaloids and therefore doesn't have that bitter taste. We've also looked at chickpea, field pea, and we've looked at different characteristics in terms of grain quality traits. We're looking at things like the seed filling within peas, and similarly, we've also looked at characteristics like seed thickness to understand how that will improve processability as we're using these types of products now in food products more commonly. I've mentioned a few times allergens, and so our lab has been really focused in on both gluten proteins, which are responsible for celiac disease, but also a number of food allergens that exist within many of the legumes. Most people will be familiar with peanut as a major allergen, but then we also know that many other legumes contain similar allergens, and so we have been working to characterise those. Within the Australian Research Council Centre of Excellence for Innovations in Peptide and Protein Science, which is quite the mouthful, we're a member of that centre, and we deliver to the Discover theme, and that's where we're looking to identify bioactive peptides from agricultural crops, from food waste, and also from food byproducts. And you can see some of the students and postdocs who've been involved with this project. This is just an example where Angela has actually undertaken bioinformatic analysis across around 200 different food crops to identify which ones have the greater propensity for delivering bioactive peptides by mapping these out in their genomes. And you can see all types of different functionality from antibacterial through to ACE inhibitory activity, anti-hypertension, and we can see which ones of these crops may be important. And then we also, at the same time, as we're looking at the potential for these to deliver these bioactives, we also look at the impacts on plant development, how these bioactives change under stress response, and then what's present in things like food waste. And so you can see, as I've mentioned before, 200 high-resolution plant genomes and variable numbers of bioactive peptides from a few hundred up to several thousand. And then we are able to then identify those that have the greatest propensity for different bioactivity, and then we can focus our interest there. And so just as some examples, you can see many of these legumes are rich in anti-hypertensive activity, anti-cancer, anti-angiogenesis, and also contain some of the cyclic peptides, which are quite unique in terms of their stability profiles. And so we've focused our interest in and around many of those particular crops. We're also applying different machine learning aided bioactive peptide discovery pathways. This is Ken's PhD work, and so looking at screening the genomes for those that have the highest antioxidant potential, taking them through into discovery proteomics so we can understand what is actually present within those grains and plants, and then developing a new machine learning model that allows us to classify these antioxidant peptides. And then we can again focus our interest on those ones with the highest potential and take them through to clinical or at least in vitro validation. So we've been applying these approaches to many different food processing byproducts. You can see here things like the spent grain that's left after brewing, or to the mango peel and seed, which are the byproducts of production after you've removed the flesh. And also things like the cucurbits, where we're looking at things like peel and seed, and these includes things like pumpkin, cucumber, and else. And then more recently, we've been looking at some of these fungal bioactive peptides and also the enzymes that are released, because fungi are highly enzymatically active and can actually produce enzymes capable of modifying foods in a way that can enhance the nutritional qualities. And so these are examples of both PhD and postdoc projects that are occurring within our lab. But with that, I am now going to pause here and hand over to Claire, who will take you through the work specifically around barley. But thanks for joining.
All right, I'm just now going to set up the slideshow for mine. Sorry, excuse me. So Luke, can I just check, can you see that slide?
Looks good. Yep.
Okay, perfect. All right, thank you. And hello again, everyone. So today I will be sharing findings from my PhD, where I use proteomics to understand the protein level changes during barley germination in malting and how these relate to water use efficiency and malt quality trade-offs to inform breeding strategies. Okay, so barley is a preferred grain for brewing because, as we all know, it has a large starchy endosperm that can be converted to simple sugars for fermentation. But these reserves are locked in the endosperm. So just get my laser up in a protein cell wall matrix. And you can see here shown in the schematic. So this is where we use a process of controlled germination or malting to mobilize those reserves. So in malting, we begin with a steep where the grain is submerged in water to initiate germination. This is followed by germination where enzymes work to break down the endosperm. And then we finish with kilning where we apply heat to stop germination and stabilize the grain. So we start with a raw hard grain and we finish with a soft friable malt. And this level of degradation is referred to as modification and is a key determinant in malt quality. The challenge is that malting is really resource intensive and it uses large amounts of water and energy. So we're always looking at ways for small efficiency gains that can make a big difference for sustainability. So this project stemmed from some findings at Pilot Malting Australia, where IGB and InterGrain breeding line achieved a similar level of modification to our control variety Flinders, but with less water. So IGB 1467 therefore offered a unique opportunity to explore what differences were happening between the phenotypes. And we asked what proteins or enzymes drive this phenotype, how do they shift over time between the genotypes, and how can this inform breeding for better malting efficiency? So to explore this question, we use a quantitative proteomics workflow. So now if genomics tells us what could happen, proteomics tells us what is happening. And the really cool thing about proteomics is that it lets us know what's going on in the grain at a given time. So it was perfect for looking at what changes were happening during malting. So both genotypes were pilot malted at Pilot Malting Australia to comparable levels of modification and sampled every 24 hours throughout the process. We then extracted the proteins, we digest these proteins into peptides, and we separate them by liquid chromatography before measuring them on the mass spectrometer. So the resulting data is then analysed with bioinformatics, and we focused on the first 4 time points to track changes in protein abundance in the early stages of malting. This gave us a dynamic protein map for each genotype. So across the time course, we identified more than 3,400 proteins, and of these, around 800 were differentially abundant. So this means they significantly decrease or increase between the time points. So we then performed hierarchical clustering with functional annotation. This is where the proteins are clustered based on their relatedness, and then we assign a biological function to that group of proteins. So this heat map shows IgB and then Flinders, and then the time points where red indicates an increase in abundance and blue is a decrease in abundance. So we're looking to see if there's any patterns in our data that jump out that could answer our biological question, what drives this phenotype? So our first question was whether there was an obvious cluster to explain the low water uptake in IgB 1467. And we found this cluster of transmembrane proteins that include aquaporins. And you can see that it was less abundant in IgB 1467 compared to Flinders. So aquaporins are a class of proteins known to be involved in water movement. And this suggests that reduced aquaporin abundance may be a factor contributing to the lower water uptake. So to confirm this, we carried out targeted proteomics. This is using multiple reaction monitoring. And we quantified 7 aquaporin proteins. 3 were plasma intrinsic proteins or PIPs, which facilitate movement across membranes, and 4 were toneoplast intrinsic proteins in the vacuole membrane that regulate water uptake in cellular turgor. Both TIPs and PIPs were lower in IgB 1467, and significantly lower was the TIP3 known to be involved in water uptake. So this supports our discovery findings that lower aquaporin abundance in IgB 1467 may contribute to its reduced water requirement. Next, we asked how IgB 1467 maintains germination under reduced water, and to explore this, we mapped the differentially abundant proteins involved in the germination metabolism. In this figure, the first 4 circles are IgB across the time point, and Flinders are the second set. And again, red is an increase and blue is a decrease. Well, there is a lot happening here, but the main takeaway is that we saw an increase in pyruvate decarboxylase and alcohol dehydrogenase. These are 2 enzymes central to a fermentative metabolism, which supports ATP production under low oxygen conditions. So this suggests that IgB 1467 shifts to a fermentative metabolism to sustain germination during steeping. And when we look at this in the context of malting, during that first 24 hours in steeping, the grain is submerged and oxygen becomes limited. And despite aeration and a period of air rest, the grain effectively experiences a transient waterlogged state similar to flooded rice systems. So to interpret this response, it helps to consider how plants cope with low oxygen. And in rice, there's 2 broad submergence strategies described. The first is the low oxygen quiescent strategy, where the metabolism slows to conserve energy. And the second is the low oxygen escape strategy. This is where growth is maintained, enabling the coleoptile to reach the surface for oxygen, but this does require more energy. When we compared coleoptile elongation, we did see that IgB had greater growth and compared to Flinders with a more quiescent-like. So while this is not rice, the phenotype suggests that IgB 1467 adopts more of an escape-like strategy, remaining metabolically active during low oxygen in malting. So when we look at the pathways underlying this response, under low oxygen, mitochondrial respiration becomes impaired and ATP production drops. In response, the grain activates hypoxic signaling through ethylene response factors, which bind to hypoxia response promoter elements and upregulate the fermentative metabolism. So both genotypes show a reduction in ATP synthase, confirming that they sense low oxygen. However, we can see that IgB 1467 has a higher ethylene or stronger response, and this is consistent with that increased abundance of fermentative metabolism. So at first, this looked like a win for sustainability, but there was a catch. Under low oxygen, continued metabolic activity causes a rise in reactive oxygen species. So to better understand how this was regulated, we look to, so in future, we're going to try and hopefully look more at the phosphoproteomics related to this, but we're not there quite yet. So reactive oxygen species, as we know, is a natural part of the germination metabolism, but under stress, excessive accumulation can cause damage to lipids, proteins, and cellular components. So both genotypes show an increase in antioxidant activity, including superoxide dismutase, which converts superoxide to hydrogen peroxide. Hydrogen peroxide is then detoxified by peroxidases and catalases, and this is assisted by aquaporins, specifically PIPs, that help move hydrogen peroxide across membranes. The high metabolic activity pressure of IgB 1467 generates more ROS. At the same time, with a lower aquaporin abundance, may limit hydrogen peroxide transport and reduce catalase-mediated detoxification. This creates a dual burden of more ROS and less clearance, leading to sustained oxidative pressure and potential non-enzymatic lipid peroxidation. So looking back at the heat map, we can see that aquaporins do cluster with cellular detoxification and response to reactive oxygen species. So this does highlight the dual role of aquaporins in both water transport and ROS regulation. So with fewer aquaporins, IgB 1467 has reduced water uptake through lower tips and reduced ROS clearance through lower pips. And this creates a combined constraint linking water efficiency directly to oxidative stress with potential impacts on malt quality. So these molecular changes have clear functional consequence when we link them back to malt quality. And in IgB, although we achieved modification with less water, we saw clear quality trade-offs. The high metabolic activity and growth are reflected in the compositional data where IgB 1467 shows lower free glucose, higher beta-glucan, and elevated MDA levels indicating increased lipid peroxidation and oxidative stress. And these changes translate to lower diastatic power, higher warp beta-glucan, and potential impacts on flavour like rancidity from the MDA. Importantly, the Colback index and friability were acceptable, suggesting that overall protein solubilization is not a limiting factor. So while we gain water use efficiency, we did lose some key quality traits. So this wasn't the outcome that we had wanted. It provides valuable breeding insight in that it helps define what to avoid and what to optimize moving forward. So it's already well established that water uptake influences modification and malt quality, and that lower water often leads to reduced quality. What this study adds is a protein level insight into how that relationship may be biologically mediated. Rather than a single driver, we see a connected system involving water uptake, oxygen limitation, metabolic strategy, oxidative stress, ultimately influencing malt quality. Within this system, aquaporins emerge as key players linking water transport and stress response. So this work moves the discussion beyond observing an efficiency quality trade-off to understanding the biological basis of that protein level. And that shift opens the door for more targeted breeding strategies. However, this does remain foundational work and we still need to validate this across a broader germplasm. So what does this mean for breeding? So from a proteomics perspective, we identified candidate protein systems, particularly aquaporins, and barley contains a large aquaporin gene family with natural variation both in sequence and expression. And that variation likely reflects underlying genetic differences such as regulatory or copy number variation, and may influence the protein abundance during germination. So the next step is to test whether the variation at this loci correlate with malting performance across broader breeding populations. And if those relationships hold, aquaporin-associated loci could become useful targets for market development. This highlights the value of applied proteomics not as a breeding tool alone, but as a way to identify candidate proteins, link them to phenotype, and guide where to look in the genome. So going forward, the goal is not to select for reduced water uptake, but to balance water transport, metabolism, and stress to maintain both efficiency and quality. So in summary, our findings provide protein level insight into the biological basis of the efficiency quality trade-off. This is foundational understanding and lays the groundwork for breeding strategies that balance sustainability with malt quality. So in closing, I would like to acknowledge that this was an industry PhD, and I'm grateful for the contributions of CSIRO, InterGrain, Edith Cowan University, and Pilot Malting Australia. This collaboration allowed us to connect proteomic data to real-world malting performance, linking research to industry application. I'd also like to thank my supervisors, especially Michelle, and to the ECU Proteomics team, and thank you for your time.
Okay, thank you. Malcolm can confirm. I know there were some issues about the chat earlier on, but maybe I could ask a few questions just to get things going. Thanks a lot for the talk. Really interesting. Raises lots of questions. Could you just give a little bit more detail in the actual basic phenotypic difference between those two lines? There's a difference in water uptake, but not in the time it took to reach to get to the right amount of final water uptake. I mean, because clearly you've got a difference in germination by 72 hours. So I'm just wondering what the.
So from what I understand from Pilot Malting Australia, so this grain was provided to me. So what they found was that so they micromalted it initially, and they found that it just modified too efficiently, which was obviously an issue. So then what they did was they did a pilot malting experiment and reduced the amount of water. So I think it was around about 2% less water was needed. And they were able to also reduce the steep time a little bit.
Oh, okay. Okay.
And that did help sort of slow the modification, but it's still very much modified very quickly. Does that answer your question?
So, yeah, I think so. Because it's potentially affecting the rate as well as the total volume of water uptake.
The other advantage is that it reduces kiln load moisture as well. So you've got another saving there on the end. But I mean, if you're losing malt quality for that, I'm not sure that it's a fair trade-off. But it was just such a unique phenotype just to be able to see. I think what IGB 467 provides is an extreme. So you can sort of use it as a way of looking back at, what we kind of don't want rather than what we do want.
But the potential to reduce steeping time or the number of steeps is potentially a huge saving, right? I mean,
absolutely. And I think highlighting also that water uptake, if you could increase aquaporin abundance, maybe you could reduce time, which is also, like you say, a saving. So there's efficiency in doing that as well. So there's advantages.
I guess the amount of water is, I guess, in Australian perspective, is a major concern. I have a feeling here it's less with maybe some modern technologies of recycling through OptiSteep and other technologies. There may be less pressure on actual total water consumption, but time certainly would be a major issue.
Well, that's the other thing. If you could increase water uptake more efficiently, maybe you could use a single steep. I don't know. I mean, that would be something that you would need to optimize or even look at a bit further. Yeah. Admittedly, I'm not a maltster, so I don't know.
But I mean, I imagine the steep regime presumably wasn't quite optimized for your IBG B1467, right? So you're seeing it reacting to more standard conditions.
Yeah, and it also, I think they reduced the temperature as well to help.
Wow. Okay.
There's a question in the chat. Luke, I don't know if you can see that.
Okay, right, great. Yes, from Christabel. Yes, she's asking whether do you think increasing aquaporin abundance could further reduce oxidative stress and increase malt quality in that way too?
That's a really great question, and I believe so. Obviously, different aquaporins have different roles. Like I said, the pips are more the plasma intrinsic proteins, so they would be more involved in managing oxidative stress. So as long as you're looking for your specific tip isoform, then yes, I do believe. But I think if you increased a tip, you would not necessarily gain the advantage of increased efficiency for the oxidative stress management. I hope that answers your question, Christabel.
We have another question from Robbie. Does Robbie, can you actually answer that, or it's a long question? That's under the power of Malcolm to.
I can see Robbie's question here. It's a really great question. It's a challenging question, like you say. So, yes.
Shall I read it out? I'll read it just in case.
Yeah, sorry.
People can't read it. No, yes, right. You mentioned the aquaporin gene family has many members. How do you propose to identify which aquaporins are potentially involved in the observed differences between IBG 1467 and Flinders? And is the phenotype quantitative? And are you looking for, following on from that, biparental or GWAS populations to understand this? So
yes,
take it away.
That's a challenging question, but a good one. Yeah, so essentially we're going to get the protein sequence, and so then we reverse, we go backwards. So we will then find the protein coding region within the genome, and then we can obviously develop markers or also go back to see if there's any SNP data around where we're located. Then it would be a case of then looking at that, associating that abundance, whether there's a copy variation or what's going on there, and then associating that with the phenotype. But that would require looking at a much larger breeding population, which we haven't had an opportunity to do yet. But yes, that would be the next steps. I haven't gone as far as looking at biparental or GWS. And the other thing I think is also a challenge with the aquaporin is that we don't know how stable it is. So who knows whether it's affected by environment or genotype. So there's a lot still to do around it to try and make it more of a stable marker, but it's still, I still think it adds value, and it's a potential candidate. I hope that helps.
There's a following question from Robbie. Said, do you know if there's a correlation between specific aquaporin family members and transcript and I suppose protein abundance? I mean, what's the relationship between the genome and what you actually see in the grain in terms of protein?
I haven't gone as far as to look at that. That's a really great question, and it absolutely is something that we should look at. But no, I haven't looked at any transcript abundance or anything like that. No. Thanks, Robbie.
Nice talk. Nice talk. Well done. Yeah.
Thank you.
From your talk, it looked like the lower tips were having a positive impact on water uptake, but your lower pips were impacting on your oxidative stress. So presumably the ideal thing would be to produce one but not the other, and presumably they're not genetically linked. So presumably that is potentially could be tractable through breeding to pull together the optimal combination, right?
Oh, absolutely. It's all about balance. I think there is efficiency savings to be made. But again, you have to trade that off with, what are your acceptable malt quality parameters or specifications that you're willing to trade it off with. But yeah, I think there's absolutely room there. It would be great to look at a bigger breeding population and see how aquaporins are working and if that phenotype does correlate. Yeah.
Presumably InterGrain have material in the pipeline, right?
They do, but I think funding's always the thing, Timbo. Like, it's great, you can have these great ideas, or, you know, maybe I should make a grant application or something. But yeah, I would, it does interest me. I am super keen, but yeah, it's just a case of, yeah, making it happen, which is the challenge.
Okay, another question from Christabel. She's wondering if you had, do you think if you had too many aquaporins, water uptake could be too quick, and potentially you could get some damage or associated problems?
I would be speculating here. I don't know for sure. I haven't read the literature on this, but I'm assuming too much water uptake again would maybe cause more oxidative stress, because essentially it would probably increase waterlogging, stress, all that. No, I'm sorry, Christabel. I'd be making incorrect inferences without correctly looking at the literature. I don't know, but it's a really great question, and another one I would love to follow up on.
Oh, Robbie's got another question. He's wanting to know what other differences there are between IBG 1, 4, 6, 7 and Flinders, and basically how closely related are they? I mean, have you got those sort of data?
Yeah, I would need to check it out with InterGrain, but I do believe Flinders is related to IGB 1467. They did have similar protein content. I think IGB, they were slightly smaller, so whether they were more steely, I'm not sure. Again, I would need to go back to the data for that. I can always get back to you if you like, Robbie, but I don't know the finer points of the actual phenotype.
Yeah, potentially a slightly smaller grain could affect water uptake as well, right? Yeah.
True.
Could I ask a technical question? How did you find the identification of the peptides given genomic information? How does that relate to your protein database? How does that relate to your identification of peptide fragments, and how comprehensive do you think that is?
Yeah, no. So your database is in proteomics. Your database is of utmost importance. So it is your go-to thing. So I used Morix. I initially tried to use the parent genome, but the thing is that didn't work with our bioinformatics tools. So I went with the Morix genome. And so what I did was you create a database, and then when you upload it to the software, it creates sorry, this is protein sequences as well. This isn't genomic sequences. And it creates an in silico library where it breaks the proteins up. So we say, sorry, it's going to get a little bit technical here. So we say we digested this protein with trypsin. So in silico, it breaks up the protein sequence into peptides. And then what it does is it matches those peptides within the program. It uses an algorithm. It's very computer intensive, and it matches those peptides. So we're looking for at least, so to identify a protein, we need at least 2 peptides within that protein sequence to say that, yes, we have this protein. So that's essentially how we do it. And then to find the genes, we obviously then I would just blast it back to the Morix genome.
So what sort of percentage of fragments were you able to identify? I mean, give a protein identification to?
Oh, the in silico library is massive. Yes, Michelle would be better suited to answer this question. Thank you, Michelle.
Hi there, and
Hi.
sorry to not be with you earlier, just listening in. So as Claire's described, we basically use the genome and any other protein databases, and we identify the proteins on the basis of the peptide evidence. As Claire said, minimum of 2 peptides is kind of expected, but we go beyond that. Generally, we achieve, if it's a high abundance protein, we might get something like 80 to 90% sequence coverage, so you can be really confident. But when we're talking about those unique peptides that that we are identifying that minimum of 2, then what you actually are doing is you're collecting peptide evidence that is unique to a particular protein isoform. And so you know that you've got a particular one based on that sequence being essentially a characteristic fragment. And so we collect that data, and depending if, as I say, if it's a low abundance protein, you may not get that level of sequence coverage that I described, around 80–90%. You may get less. And certainly in the case of some proteins that we're very familiar with, which is the gluten proteins that are there, they have regions that are really hard to sequence and characterize. So we know that we get lesser sequence coverage across them, but we aim for those really unique marker peptides. And then we collect that data and do the quantitative analysis across. And I think the description you had earlier around the importance of, I guess, linking back to transcriptomic data, just to take us back there for a minute, is also that we can select peptides that are specific to different isoforms, and then we can say, okay, well, how do they vary across genotypes? And then we can start to understand the importance of those as it relates to how the phenotype presents. The challenge, of course, we have here is that we were looking at malting barley, and we were trying to do it in an industrial setting. So we were having, our batches are typically 80 to 100 kilos of grain at the time. And often for breeding wines, we don't have access to that volume of material to be able to characterize those malting traits and characteristics at that, in that industrial setting. And so we then have to look at things like micro malting and other techniques. So yeah, it was a really interesting study, but expanding out at the level that we did it at to breeding wines wouldn't be possible in that way because of those sort of the limitations on breeding material. But there are approaches that you can use with micro malting that would allow you to access more genotypes and then be able to correlate better those individual protein isoforms back. So hopefully that kind of answers a couple of questions that were in the chat.
Cool. I was just wondering, do you have any feeling how much of an issue is it that you're looking at Flinders and IBG 1467 through a Morex genome protein lens, if you like? I mean, if there's particular copy number variants or things that are critical for the differences here, if you're looking at it through a Morex lens, does that limit what you can, to some extent, what you, for fine detail, you can discern here?
Did you want me to jump in on that, Claire, as well?
Yes, please.
Looks like it, yes.
Yeah. Yes, it absolutely does.
Okay.
But there is there is a lot of conservation between the barley variants, but we know we'll be missing some information. So that means that in the case of an individual protein, you will capture regions of that protein, so your sequence coverage will be less than it would be if you had a matched genome to genotype. So yeah, there will be missing data, but there's generally enough to be able to characterize most proteins.
So can I ask what's the next step? You're saying this was your PhD. Claire, what's next in the pipeline?
Next in the pipeline. So I'm hoping to look, I don't know whether you picked it up in the presentation, but I'm hoping to look at phosphoproteomics. I managed to secure a small grant to look at method development and optimization for phosphoproteomics in barley. So I'd like to really understand that signaling and see if, like what I said with the low oxygen sensing, and whether we do have that the low oxygen escape strategy present in IGB 467, because I think maybe that's also an avenue. I know that so modification efficiency could also be something if we can see how that works. But yeah, again, so with phosphoproteomics, we're looking at phosphorylation events. So we're looking at switching and stuff like that. So I'm not there yet. Again, like I say, I've just got the grant, so it's about, I still need to optimize those methods. But yeah, that's where I'd like to explore.
Good luck with that.
Yeah.
Yeah, just want to check if there's any more questions coming through on the chat. Oh, hang on. There's another question from Robbie. There are lots and lots of tissue-specific, genotype-specific, and temporal transcriptomic data in the public domain that could be assessed online. Have you explored whether there's using that information could point to your priority targets?
That's a great idea. Admittedly, no, I haven't looked at that data. I will make a point of that's something that I definitely need to explore. But no, at this stage, I haven't looked at that. Thanks, Robbie.
Any other questions? Any comments? In which case, I think we've come to an end. Just want to thank you both very much. Thanks, Claire, for your presentation, and Michelle as well. And thanks for the work and the answers to all the questions. So thank you, and hope to see you again. Cheers.
Yes. Thank you to you too. Thank you for the opportunity, and thank you everyone for joining online. It was a pleasure. Thank you. Take care.
Thanks all.
Do I just leave now, Luke?
I have no idea. I'm not in charge here at all.
That's us finished. So yeah, that went really well. Thanks very much.
That was good. No, it was good timing as well. About an hour is about fine. So,
Okay,
no, thanks
perfect.
for that. No,
All right, thank you for your time.
No, that was really, really interesting. And there's a lot of I know Sophie's Christabel's a PhD student in Nottingham with working on water uptake. So I think she was very interested in some of what you're putting out. So that's.
Well, I might reach out to her and see if she would like to chat anymore. But yeah, no.
Yeah, absolutely.
It was great. Again, this is just one small story as well because there's so much going on. But yeah, thank you very much, and have a lovely time and enjoy.
Right. Yes.
That's great. Thanks,
good to see you. Thanks.
Thank you, Malcolm.
Thanks again.
See you, Luke. Bye.
Thanks.
Okay. Cheers.